Hello dear students, today we are going to discuss about the third type of bacterial reproduction method, that is transformation. So it is one of the most simple and important topic which will be asked for your examination. So as we know that before that lecture we already discussed the two methods of that bacterial recombination or sexual method of bacteria, that is conjugation and transduction method. So last one, third one method is transformation method. So we will discuss here about the next method, that is transformation of bacteria or transformation in bacteria. So as we know that here in transformation, first we discuss about how the transformation takes place. Only within 3 to 4 steps the transformation process is completed. That transformation method was first described by the scientist Griffith in 1928. So this is asked for your objective type of question. Transformation method was first described by the scientist Griffith in which year? In 1928. Okay. The next we discuss about the steps involved in transformation process. For your examination, if the question is asked, write about the transformation in bacteria, so you can write the given step which is mentioned in bold. Here it is mentioned in slide also. Okay, so you can draw the diagram and write the description about that diagram which is given in PPT. So first we discuss about the definition of transformation. So transformation means what? There is a transfer of DNA into the bacterial chromosome, and once the DNA or transfer of DNA from one bacterium into another bacterium by the dissolution of the wall. Okay, so after the entry of DNA inside the bacteria, so there are two types, the successful transformation and unsuccessful transformation. So we will discuss in later what is successful and unsuccessful transformation. So let us begins the steps involved in transformation method. So in first step of transformation method is donor DNA fragments binds to protein on the cell surface of bacteria. So as we know that donor DNA from the other bacteria, it is double stranded. It is mentioned here, double stranded donor DNA is present, and that donor DNA during transformation process, that donor DNA comes in contact with the cell surface of bacteria. Okay, and as we know that the cell wall of bacteria is made up of the or lipids and polysaccharides and proteins. Okay, so that donor DNA fragment or double stranded DNA that binds tightly to the proteins which is present on the cell surface. Okay, so this is bacterial cell, as we know that it is indicated in diagram. The next one, it is chromosome, or it is called as a bacterial chromosome, which is present inside the bacterial cell. So what happens in first step? Donor DNA fragments or donor DNA, double stranded DNA fragments that binds tightly to the proteins which is present on the outer surface of the cell wall of the bacteria. Next, in second step, what happens? That donor DNA fragment is transported inside the host cell. Okay, so when that donor DNA comes in contact with the cell wall or outer cell of bacterial cell, so the dissolution of that cell wall takes place, and that donor DNA which is to be double stranded, so they enters inside the bacterial cell. So when they enters inside the bacterial cell through the host surface or through the bacterial surface, so it becomes single stranded. Okay, so that's why the second point is given here. Donor DNA fragments, that is double stranded, is transported into the host cell, that is they enters inside the host cell, and it becomes single stranded. And third one is there, once that donor DNA, when they enter inside the bacterial cell, there are two possibilities. So first possibility is that either that donor DNA they deposited over the bacterial chromosome. When they deposit or incorporated when the donor DNA is deposited or incorporated over the bacterial chromosome, then it is called as a successful transformation process in bacteria. So what is successful transformation? When single stranded donor DNA fragment is incorporated into the host chromosome or into the bacterial chromosome, then it is called as the successful transformation. Okay, and sometime what happens, sometime once the DNA is enters inside the bacterial cell, then instead of incorporation into bacterial cell by the donor DNA, it becomes break up into the small pieces. So when that donor DNA fragment it's break up into the small pieces or small fragment, then it is called as an unsuccessful transformation. There is no deposition of that donor DNA, single stranded donor DNA, into the bacterial chromosome. When such a condition is occur, then it is called as an the unsuccessful transformation. So let us see again, let us recap. We review what happens in first step. Donor DNA fragments binds to proteins on the cell surface at first stage. Second, that donor DNA from another bacteria, that fragment is transported inside or into the host cell, and when they enters inside the host cell, it becomes single stranded. Okay, and third one, once they enters inside the once that donor DNA they enters inside the enters inside the bacterial cell, then it becomes single stranded and they are directly incorporated into the host chromosome or bacterial chromosome. When such things happens, then it is called as an successful transformation. And when that donor DNA fragments is break up into the small pieces without deposition over the bacterial chromosome, then it is called as the unsuccessful transformation. So this is about the transformation process in bacteria. So if for examination, if the question is asked, describe in brief about transformation process, so you can write the diagram along with the description which is given in that diagram. So please refer that PPT for your semester end examination also. Okay, otherwise I also send the PDF of that 3 sexual method, that is conjugation, transformation and the transduction, that we already sent to on your WhatsApp. So the remaining 2, conjugation method and transformation method, regarding that topic I will send the PDF or PPT of that both topic. So if you have any difficulty regarding the conjugation process, transformation process and the transduction process, so you can call me or personally WhatsApp me, whatever your queries is there. Okay, so there is the end of that topic. Thank you.